Hello students, in our last lecture we have discussed about Rous sarcomavirus and how this particular type of RNA virus is creating oncogenesis, responsible for oncogenesis in human. Then we have discussed about proto-oncogene, tumor suppressor genes or anti-oncogenes and how their mutation is actually changing the normal cell into cancerous cell. Now today we are going to discuss about another type of virus, that is hepatitis virus, responsible for hepatocellular carcinoma or liver cancer. It is believed that the chronic infection with hepatitis B virus is the most common risk factor for hepatocellular carcinoma or liver cancer. Hepatitis B virus is indeed a complex virus because it occurs three different morphological forms in serum. And here you can see in the first slide, these are the three different morphological forms. The smaller spherical one and the tubular one, or the helical one, which is the elongated form, they are generally the non-infectious form. Whereas the larger spherical particle, that is, you can see in the picture, the left-hand side, this is actually the infectious particle and actually known as dense particle. So here you can see the smaller spherical particle with an average diameter of 22 nanometer and the filamentous or tubular particle that is also having the length of around 22 nanometer in diameter. They are the non-infectious form of hepatitis B virus, whereas the largest spherical particle, that is the double-celled DNA virus, that actually replicate in liver cells known as DEN particle, and this particular virus have a diameter, spherical virus have a diameter of 42 nanometer and considered to be infectious. So here you can see the general perception is that whether all the forms of hepatitis B virus, all the three forms of hepatitis B virus are infectious. So the first two forms, as I said, the first two forms are non-infectious. But the third form, that is the DEN particle, they are actually the infectious particle. And they represent the intact hepatitis virus. Most frequently observed particles are the smaller spherical particles and the tubular particles, and the DEN particle, they are the less frequently observed particle. As I have already mentioned, they are double-celled, they are spherical, enveloped virus or enveloped structure with a diameter ranging 42 to 47 nanometers. And they have three different antigens. The outer surface means the envelope, they contain a particular antigen. That particular antigen is known as surface antigen, this HBsAg. They have three different forms of the viral envelope protein: L form, M form, and S form. And just under this, they have the capsid, and the capsid belongs to the symmetry of icosahedral symmetry. And this nucleocapsid is a nucleic acid that contains a partial double-stranded DNA. And this nucleocapsid maintains the icosahedral symmetry as a diameter of around 27 to 28 nanometer. So apart from the HBsAg antigen, they have two other type of antigen. One is HBcAg, that is known as the core antigen. Another one is a HBeAg, that is pre-core antigen. So within this, they have, within the capsid, that contains the genome, that is the partial double-stranded DNA. with the length of 320 base pair, and they are in circular configuration. One strand of the incomplete double-stranded hepatitis B viral DNA is regarded as the positive strand. The incomplete strand is the positive strand. And this positive strand is associated with an enzyme that is the viral DNA polymerase. This gives an appearance of partially double-stranded and partially single-stranded DNA. Here you can see some overlapping genes there, and these overlapping genes are 4 different overlapping genes are there: S gene, C gene, X gene, and P gene. All the genes are depicted here with different colors. So C gene in blue, red gene, the longest gene, P gene is the longest gene marked in red, then S gene and X gene. So hepatitis B virus have 4 different genes, and these 4 different genes, they are the overlap genes. The genome is very compact. All the genes are residing with 3.2 kilobase pair of DNA length, and they encode 4 different proteins that are actually functioning as antigens. So what are the 4 different proteins? The S gene, they have the 3 different regions rather. One is pre-S1, pre-S2, and S. This particular 3 different regions of the S gene, they actually synthesize the antigens, that is the major antigen S, middle protein M, and large protein L. And all these 3 proteins, they actually govern the surface antigen protein, that is HBsAg. The C protein having 2 regions, pre-C and C, this all together governing HBcAg and HBeAg antigens. And the largest gene, they actually encodes or synthesize DNA polymerase, reverse transcriptase, RNase H activity, and the X gene is for hepatitis and responsible for HCC, means hepatocellular carcinoma in human. Now, genotyping is very important, for it has a clinical significance. Determining the genotype could be helpful for predicting the outcome of the disease. And definitely, it gives some idea how to develop antiviral drugs, means how to develop antiviral therapy in patients with chronic hepatitis B infection. Genotype B appears to be associated with less rapidly progressive liver disease and cirrhosis of liver, or delayed appearances of hepatocellular carcinoma than the other 2 genotypes, that is C and D. Patients with the infection by genotype A are more likely to clear circulating viremia and to achieve HBeAg and HBsAg seroconversion both spontaneously and in response to antiviral therapy. So from this, we can conclude that genotype B is more dangerous than the other 3 types, genotype C, D, and P, A. So what is actually HBsAg? This is surface antigen of hepatitis B. This particular antigen was first discovered by Bloomberg et al. In 1965 from the native Australian patient. That is why it is also termed as Australian antigen. This particular antigen contains a group-specific antigen A with 2 pairs of mutually exclusive subdeterminants, D or Y and W or R. So 4 phenotypes of HBsAg is available. Those are ADW, AYW, ADR, AYR. And Information regarding this four phenotype of HBsAg is very useful in the epidemiological investigation of HB hepatitis B virus infection. Hepatitis B have three different carriers: simple carrier, carrier, simple carrier, and supercarrier. A person with detectable HBsAg antigen in blood for more than 6 months are called normally a carrier. And if the person having HBeAg negative but HBs positive, and the titer value is less, that is present in his or her blood, then large volume of blood is required for transmission, and here the infection is less, means low infectivity in case of simple carrier. So who are the super carriers? So HBeAg means pre-core antigen positive, highly infectious, means 0.00001 ml of plasma can transmit infection. Here, in case of simple carrier, large volume of blood transmission can transform infectivity, but here a very negligible amount of plasma can transmit infection. And if we go for serological test of the patient, taking blood from the patient, and then definitely we can get elevated transaminases there. So here we can notice high infectivity, and these are the super carrier. Now we are going to discuss about the mode of transmission, how this particular virus invade into human. Mostly hepatitis B and hepatitis C virus, they transmit, they invade in our body, or they transmit through the parenteral mode, or else through blood transfusion. And here, chances of transmission by the contaminated needle pick injury in case of HBV is 30%. We have a general perception that HIV is very contagious. And if a person undergo needle pick with a contaminated needle, then the chance of contamination is nearly 100-fold more than HIV in case of HBV. So in case of HBV, it is 30%, in case of HCV, it is 3%, and in case of HIV, it is 0.3%. Again, contaminated blade, use of contaminated blade can also transmit HBV. Sexual intercourse can also transmit HBV. And from mother to child, it can also be transmitted.